Good day, everyone. I'm Simya. Today, I will present an article titled Deep KEGG, a multi-omics data integration framework for cancer recurrence predictions and biomarker discovery. This article is related to deep learning, multi-omics, and bioinformatics. The article I selected is titled Deep KEGG, a multi-omics data integration framework with biological insights for cancer recurrence prediction and biomarker discovery. It was written by Lin, Liao, Chen, Zhu, Pan, and Chen, and published in Briefings in Bioinformatics in 2024 by Oxford University Press. I chose the article because it applies deep learning to integrate multi-omics data and also provide biological interpretation. This is important because many deep learning models can achieve high accuracy, but they are often difficult to explain. Deep KEGG tries to solve this by using KEGG pathway knowledge, so the model can show which genes, mRNA, and pathways are related to cancer recurrence. For the input data, the study used two major public cancer databases, which are TCGA and Target. From TCGA, the authors used four cancer datasets, which are breast cancer, liver cancer, bladder cancer, and prostate cancer. These datasets include three types of omics data, which are SNV, mRNA, and miRNA expression. From Target, the authors used two pediatric cancer datasets, which are Target AML and Target WT. These datasets include RNA expression and miRNA expression. The target label is binary classification, which means the model predicts either recurrence or non-recurrence. Deep KEGG is an end-to-end deep learning framework with four main modules. The first module is the biological hierarchical module. This module maps genes and miRNA to KEGG pathway nodes using biological relationship matrices. This is useful because it replaces fully connected with biological meaningful connections. As a result, it can reduce overfitting and improve interpretability. The second module is pathway self-attention module. This module learns the correlation between patients and both in the pathway feature space. In simple words, the model tries to understand how similar patients are to each other based on pathway level information. The third module is classification model. The features from different omics types are combined and passed into a multi-layer perceptron or MLP to predict cancer recurrence. The final module is the interpretability module. It uses deep leaf to calculate feature importance scores, helping the researchers identify important genes, miRNA, and pathways. Now, I will continue with the classification performance. The authors evaluated deep KEGG using several metrics, including AUC, AUPR, accuracy, and F1 score. AUC measures how well the model suppresses recurrence and non-recurrence cases, and AUPR is especially useful when the dataset is imbalanced. The model was compared with seven baseline methods, including SGD, logistic regression, decision tree, KNN, SVM, PubCNN, and MogoNet. Based on the result, this KEGG achieved the best overall performance. For example, in terms of AUPR, deep KEGG achieved 0.947 for liver dataset, 0.946 for breast dataset, and 0.962 for target WT. The largest improvement can be seen in the breast dataset, where deep KEGG improved over the second base method by around 12.2%. This shows the deep KEGG is more effective in predicting cancer recurrence compared with traditional machine learning and other deep learning models. Besides prediction performance, the study also investigated biomarker discovery and biological interpretation. First, the ablation study showed that the integrated multi-omic data perform better than single omics data. This means combining different biological data types give the model more complete information. Second, deep KEGG identified important biomarkers using deep leaf importance scores for liver cancer. Examples include CYP2E1 and CXCL12. For bladder cancer, examples include ERBB2 and HSAMIR 432. These biomarkers were supported by previous literature, suggesting that the model can identify biological meaningful features. Third, the authors performed survival analysis using Kaplan-Meier curve. Some genes such as CDAA, CDC25B, SCACA, PIK3R2, CD3D, COL1A, 1 and FN1 show significant survival difference between high expression and low expression groups. This means deep KEGG is not only useful for prediction, but also useful for understanding the biological mechanism behind cancer recurrence. Although deep KEGG achieved strong results, there are still several limitations. First, the datasets are relatively small and imbalanced. Second, the model depends heavily on the KEGG pathway datasets. Third, the model does not integrate image data. Fourth, the model only uses SNV, mRNA, and miRNA data. Finally, the biomarkers were mainly validated using literature review and survival analysis, but not through wet lab experiments or prospective clinical studies. To improve the study, I suggest 5 possible improvements. First, deep KEGG can be extended into a multi-module by integrating pathology images with omics data. This is useful because images can capture tumor morphology, while omics data capture molecular changes. Second, the model can use more pathway databases such as Reactome and gene ontology instead of relying only on KEGG. Third, transfer learning or federated learning can be applied. This is useful for real cancers where data are limited. Fourth, more omics layers such as DNA methylation, CNV, and proteomics can be included to provide broader biological coverage. Lastly, prospective clinical validation should be conducted using independent hospital datasets. This is important because high performance on public datasets does not always guarantee real clinical usefulness. To conclude, deep KEGG is a strong example of deep learning in bioinformatics because it integrates multi-omics data, predicts cancer recurrence, and provides biological interpretation through biomarkers and pathway. The article is cited as below. Thank you.